My name is Jeff Tyner, and I am excited to present an introduction to the Beat AML Dream Challenge, which was supported by the NCI CTD2 Cancer Target Discovery and Development Network. I will provide a brief introduction to the current state of AML research, an overview of the Beat AML project, which supported the datasets used for this challenge, and then we will hear from the winners of the two sub-challenges. Outcomes for AML patients have historically been quite poor, and there has been little improvement over many decades. Clearly, this represents an unmet need for improved therapeutic options, and the goal of the Beat AML program, as well as this Dream Challenge, has been to optimize new therapeutic strategies for AML patients that are tailored to biological, genetic, and clinical features of disease. AML is one of the most common forms of hematologic malignancy, and the standard of care therapies that combine anthracyclines with nucleoside analogs have, until recently, remained unchanged for the past 40 to 50 years. Certain subsets of disease defined by genetic features such as rearrangement of the retinoic acid receptor, or clinical features such as age and performance status, have led to the use of alternative therapeutic approaches. Fortunately, the past 2 to 3 years have seen a series of new agents approved for AML. Most of these approvals have been for therapies that selectively target mutated enzymes in AML tumors, most notably inhibitors of FLT3 or IDH1 or 2. One of the most recent approvals has been for the BCL2 inhibitor venetoclax in combination with hypomethylating agents for elderly patients not fit for standard of care therapies. There are numerous additional agents in trials and in preclinical development, only a few of which are listed on this slide. A review of the genetics of AML reveals some of the challenges faced by the field in the development of genetic targeted therapies for this disease. Numerous studies of the somatic mutation rate across diverse cancer types have uniformly shown AML to have among the lowest somatic mutation burden. While these data are certainly accurate, they may create a misleading impression that AML is a genetically simple disease. A closer look reveals that this low mutation rate is comprised of a large diversity of combinatorial mutation patterns. This can be exemplified by assessing the combinatorial complexity of just 7 commonly mutated genes that collectively yield several dozen different configurations of mutational co-occurrence, as shown here. It can also be illustrated by mapping all of the frequently mutated or rearranged genes across large cohorts of patients, where we can observe that there are many different mutated genes, and these mutations co-occur in a diversity of different patterns. To better characterize the potential for therapeutic responsiveness in the context of this genetic complexity, we launched the Beat AML Consortium in 2013. This Beat AML 1.0 project was a preclinical effort to study primary specimens from a large cohort of AML patients using an ex vivo drug sensitivity analysis from which drug response data could be integrated with mutational and gene expression data from exome and RNA sequencing, as well as detailed clinical annotations. The majority of this dataset was published in Nature in 2018 and served as the training dataset for this challenge. One of the unique components of this dataset was the inclusion of ex vivo drug sensitivity data for a majority of the cases. These data were obtained by purifying mononuclear cells from freshly obtained patient specimens and immediately exposing these primary cells to a library of small molecule inhibitors plated as 7-point dose-response curves in arrayed format. After a short incubation period of 3 days, the impact of each drug on cell viability was measured using a colorimetric cell viability assay. All conditions were normalized to the average of a series of wells containing only vehicle, and probit curve fits were used to obtain dose-response metrics such as area under the curve. As a result, this dataset contains drug response information for most specimens in the cohort against a library of over 100 drugs. The basic dose-response data are shown here, with area under the curve values organized with unsupervised hierarchical clustering. Whole exome sequencing was also performed on most specimens, and high-confidence somatic mutations were called for each sample. In addition, chromosomal rearrangements were curated from a union of clinical cytogenetics and gene fusion calls from RNA sequencing data. Comparison of this dataset versus other large AML datasets, such as TCGA, for frequency of the most common mutations reveals good overall agreement between cohorts, with only a handful of events that show discrepant frequency, likely due to technical differences in exome capture and/or sequencing platforms used for the respective cohorts. RNA sequencing was also performed on most specimens, and unsupervised clustering of gene expression values shows groupings that align with prominent genetic subsets of disease. Notably, we also included RNA sequencing of two groups of healthy controls, one representing RNA from whole mononuclear bone marrow cells from healthy volunteers, and the other CD34 selected healthy bone marrow cells, which represents a population enriched for stem progenitor cells. All of these data can be found in the NCI Genomic Data Commons repository, and our informatics collaborators, led by Dr. Shannon McQueenie, also created a public portal called Visome that can be used to explore and visualize this dataset. In this way, we hope to democratize the access and use of this dataset, and indeed, this Visome platform has been broadly accessed over the past 2 years since its launch. We wanted to further engage the computational community to explore and investigate this dataset, so we partnered with Sage Bionetworks and, supported by the NCI CTD2 Cancer Target Discovery and Development Network, we launched a DREAM Challenge. The first sub-aim of this challenge was to optimize predictivity of ex vivo drug responses using the other genomic and clinical features of this dataset. The second sub-aim was to identify best strategies for predicting clinical response using any of the above data features, including the ex vivo drug response data itself. For each specimen in the training dataset, there were a variety of clinical, ex vivo drug sensitivity, mutational, and gene expression features, some of which were numerical and others categorical. The challenge launched December 19, 2019 and consisted of 3 phases. In the open phase, participants could develop and train their methods. In the leaderboard phase, participants could continue training their methods and could also submit their methods for scoring. Final submissions were then assessed during the validation phase, with results announced in late May of 2020. The training dataset was comprised of 213 samples, the leaderboard dataset contained 80 samples, and the validation dataset contained 65 samples. It should be noted that due to the realities of working with primary patient specimens, where cell yield is highly variable and replacement material cannot be generated for any tests that fail data QA/QC metrics, the data matrix is not 100% complete for any of these cohorts. The overall completeness was 78%, with the largest number of specimens containing mutational data, second most containing gene expression data, and a slightly smaller number containing ex vivo drug response data. This challenge was a so-called model-to-data approach in which participants submitted their methods in Docker containers that were then executed in the cloud. We did this for 2 primary reasons. First, it enables reproducible and portable generation of results. Second, it allowed us to use the ground truth of Sub-Challenge 1, namely the ex vivo drug sensitivity data, as covariates in Sub-Challenge 2 without introducing biases and overfitting due to leaking of that ground truth across the sub-challenges. 27 teams submitted methods to the validation phase of Sub-Challenge 1. Nearly all methods used expression data, with many methods using all 3 data modalities. We included 2 baseline comparator models, a standard ridge regression model, and the top performer from a related drug prediction challenge in breast cancer cell lines called Bayesian Multitask Multiple Kernel Learning, MKL, in the slide. 10 participant methods outperformed both comparators using both Spearman and Pearson correlations. A common feature of many of these top performers was the simultaneous modeling of all drugs within the dataset, as opposed to independent modeling of each. This exploits the correlation in responses across drugs. The top performer, Team AAUH from Aalborg University Hospital, used this approach and achieved a Spearman correlation of 0.37 that improved upon the Spearman correlation of 0.32 achieved by the ridge regression model. Rasmus Brunden will give more details on their approach in the next talk in this session. 14 teams submitted methods to the validation phase in Sub-Challenge 2. There was a statistical tie between 4 teams based on the primary metric concordance index. Hence, the top participant, Y. Mimari, was determined by the secondary metric AUC. Team Y. Mimari from the University of Cambridge improved upon a baseline Cox proportional hazards model that used all data modalities, achieving a concordance index of 0.77 relative to the baseline method's 0.68. Yassine Mimari will present the details of this approach in the final talk of this session. I would like to acknowledge Jacob Roberts, who led this challenge. He was supported by Verena Chung and Tom Yu, both at Sage Bionetworks, who developed and applied the challenge infrastructure. Verena is also participating in our post-competitive collaborative phase analysis, which is being led by Brian White, also at SAGE, Anna Shihoiska at the Institute for Molecular Medicine Finland, also known as FIM, and Tero Aitokallio at FIM and the Institute for Cancer Research, Oslo University Hospital. I've already mentioned funding from the NCI CTD2 program, and I want to explicitly thank our program director, Daniela Gerhard, and Justin Ganay at SAGE for championing this work. I would also like to thank all of the Beat AML Consortium partners for developing this Beat AML dataset. I'd be happy to take any questions, and I'll look forward to hearing from our sub-challenge winners in the next two presentations. Thank you. Thank you.